Asexual reproduction and mitosis. So there's going to be a lot of terminology here, and we need to make sure we know what it is that we're talking about. So genetics in general is the study of genes, heredity, so the inheritance of those genes, and the variation of inherited characteristics. We're going to be talking about chromosomes and the different forms in which they take at different points during cell replication. And of course, the chromosome itself is made up of DNA, which is this double helix of deoxyribonucleic acid, and it is the code that carries our genetic information. A gene is a portion of DNA that codes for a particular trait or characteristic, and so a certain section of that DNA is going to encode for certain information, and we refer to those, if it's a particular trait, as a gene. Haploid refers to cells that contain half of the number of chromosomes. So we're going to have points in which cells are going to be haploid or diploid. Diploid being the other one, and in this one you're going to have the two sets of chromosomes present. The number, shapes, and size of chromosomes vary between species. So, for example, humans have 46 chromosomes. Dogs actually have more chromosomes than we do. So the number of chromosomes doesn't tell you a whole lot about the complexity of the organism or the number of genes within that organism. Think of a chromosome as merely a division of the entire genetic code. So think of it as like the different volumes in which the entire genetic code is written in. So we have 46 chromosomes. This is our diploid number. And essentially what it is it's 46 chromosomes, but each chromosome comes as a pair. And so we have 23 pairs of chromosomes, which makes up our 46 chromosomes. So this is our karyotype, or when you look at our chromosomes and you match them up and to see who is paired, you look at those different pairs. We have 23 of those pairs. That would be our karyotype. So sexual versus asexual reproduction. With sexual reproduction, new individuals are produced from fusing two different sex cells. So we're having genetic information coming from both of the parents. This allows for greater genetic variability, and we know that with greater diversity, greater variability, the species itself is going to be more adaptable. If there's more variety, more diversity within that species, then that species is going to be able to survive better. And by having the different fusion of genetic information from two separate individuals, you get more variability. The problem is it does come at some cost, and so these two organisms are going to have to meet in some way and actually have those production of those sex cells. They can't just reproduce on their own. It does take two of them to do it, so that is going to have some cost to the species. Such as peacocks, if they're trying to attract a mate, works fairly well for attracting a mate. However, it can also work very well for a predator trying to catch them. Whereas with asexual reproduction, the individual can just produce a new individual. So this one individual at whatever point can just make a new individual. It doesn't have to rely on anyone else. It is just able to essentially copy itself, and now you have two individuals. Because they're genetically identical, there is going to be a cost to that as well. So yes, it is very easy to do, but the cost is that the new individual is an exact copy of the original individual, so there is no increase in the diversity within that species. Cell division, we're going to talk about parent cells. So the original cell that you start off with is the parent cell. The daughter cells are what they become. So you imagine you have an original cell, and if it splits into two, you're going to have two new cells. The parent cell is the original cell, and the daughter cells are the new cells. Now realize it's not that the parent produces those two daughter cells, it's that the parent becomes those two daughter cells. And daughter's not referring to its gender or anything like that, just that it has come from the parent cell. These are going to have exact copies of the DNA passed on, assuming this cell is just divided. It should be exactly the same, that the two daughter cells should be copies of the parent cell. And this allows for genetic continuity. So we don't always want diversity when we're copying cells. For example, if you are injure your hand and you want to replace your skin cells, you don't want some new type of skin cells, you want the same ones that you had before. And so within cell division, there is an advantage to having genetic continuity. This is carried out by somatic cells, which essentially are all the body cells except for reproductive cells. This allows for growth, tissue repair, and cell replacement, all of which you would not want to have your genes coming up with a new combination every time they went to do it. You just want to have a copy of what was already there. And this happens in essentially two parts. So that we're going to focus on mitosis, the steps involved in making that replication. And then essentially the last step is them splitting apart completely, and they call that cytokinesis. So the cell cycle can be broken up into different phases, but we're going to focus mostly on mitosis. However, mitosis only makes up a portion of the cell's life cycle. Generally, the cell, for the most part, is doing other things most of the time, but we're going to focus on the part where you actually take that original cell and it splits into these two daughter cells, that process called mitosis. Before we can get there, though, the interphase, or the period between cell divisions, the rest of it that's happening other than mitosis, is broken up into a couple of phases. So we have the G1 phase, which is essentially the growth phase. The synthesis phase, and this is where the DNA is replicated, which is going to be important if the cell is going to split into two. We have to have two copies. And then the second growth phase, or G2, again, there's a couple more things that are going to happen, but basically getting ready for mitosis. So mitosis itself, we'll look at the different stages of it, but the first one, right before it actually happens, we have this interphase, and essentially the chromosomes are irregularly spread throughout the nucleus and called chromatin. So think of the DNA is sort of all dispersed and not a particularly well arrangement. It is irregularly spread throughout, it is uncondensed, and that is referred to as chromatin. One thing to note is that we're breaking it up into separate phases, but it's not like it sort of goes and stops at each phase and the next phase starts and then it stops and the next phase starts. It's just a continuous phase, and we break it up into steps to make it a little easier to understand. All right, prophase. So in the prophase, the centrioles are going to move to opposite poles. So imagine if you have your cell, we're going to end up with one centriole on one side and one centriole on the other side, if it's an animal cell anyways. And if these are found in pairs, we call them a centrosome. So the centrosome would be the pair of centrioles, of which we'd have one at each end of the cell. The centrioles together make up a centrosome, and we have a pair at one end and a pair at the other end. The chromatin that we saw earlier is going to condense, and that is where we first see what the structure that's known as the chromosome. So we can actually see these individual bundles of DNA, and they are condensed together and packaged up in a form that we call chromosomes. They're actually visible with a microscope, and they are made up of sister chromatids. The sister chromatids that make up the chromosome are joined together in the middle with this part that's called the centromere. We're going to go over this again in a minute. So a pair of sister chromatids make up one duplicated chromosome. So during prophase, the nuclear envelope is going to break down. These chromosomes are going to have to move around. So essentially, the nucleolus disappears. The nucleus itself is not there anymore. These fibers, microtubules, form what's called a mitotic spindle fibers. And these fibers help to move the chromosomes to where they need to be in the cell. They're going to grab onto that centromere that we saw earlier. So the chromosomes are during prophase, the chromosomes are going to migrate to the center of the cell. So let's see if we've got this straight. We have a whole bunch of terminology that is pretty similarly sounding, but they mean different things. We have to make sure that we know what they mean. So your centrioles are going to be essentially these little, look like cans almost, but if you have one of these things where these fibers are going to be growing out of, we call it a centriole. If we have a pair of them, they come in sort of sets of two, the pair of them is going to be the centrosome. So one of them is the centriole. So imagine this guy right here. And if you have a pair of them, that's called the centrosome. Chromatin is DNA that is not condensed, it's just wiggly all over the place. It is spread around, and that's going to be found in the nucleus. When the DNA is being used by the cell, that's essentially what it looks like. It is DNA that is uncondensed. But during replication, this chromatin is going to have to condense into these chromosomes, and so they kind of end up looking like a butterfly almost, or like an X sort of thing. And so that condensed version of the chromatin is called a chromosome. Now, the chromosome is held together. There's two parts to it, and they're held together with this thing called a centromere. The two parts that are held together, so this one part right here is held together with the centromere to this other part on the other side here. Those individual halves are called chromatids. So we have here in prophase our centrioles right here. So imagine those like can-like things. And they're going to be associated with the spindle fibers that are going to help move the chromosomes around. So the two centrioles there making up the centrosome. And down here in the nucleus that's slowly degrading, we have these chromosomes. And so chromosomes made up of a chromatid held with a centromere to another chromatid. So the two chromatids together being held by the centromere, that is the chromosome. Metaphase, these chromosomes are going to line up around the middle of the cell, think of it like the equator of the cell, and these chromosomes are going to be held perpendicular to the spindle fibers. So what that's going to look like is we have our chromosomes, and again, the centromere is what's holding those two chromatids together. The spindle fibers are going to be attaching to that centromere on either side, but metaphase is where they all line up along the middle. And so that we have one sister chromatid on this one side, you have the other sister chromatid on the other side, and the spindle fibers are going to be attaching to those chromatids. Anaphase is where those chromatids get separated away from each other. So they're attached to the centromere, they're going to be pulled apart at that centromere, and you're going to have two identical single-stranded chromosomes. So each chromosome is pulled towards the poles as the spindle fibers shorten. So you can imagine if we have a chromosome that has a centromere here, and imagine the spindle fiber going to one side of the cell and another spindle fiber going to the other side of the cell. That chromosome is made up of sister chromatids. So the next step where they get pulled apart, so imagine the one side is being pulled and the other side is being pulled. Let me draw in the centromere here. So the spindle fibers are going to be pulling these guys away from each other towards the poles of the cell. Now, when they're attached together as a chromosome, so this is a chromosome, it's got two sister chromatids in it while they're attached. Once they're separated, this is going to be a single-stranded chromosome. So once they are separated again, we don't call them chromatids anymore. They are themselves now their own chromosome. So anaphase is going to be pulling those chromosomes apart to either end of the cell. And again, what was attached together as a single chromosome made up of two chromatids, it gets pulled apart. And now what was those chromatids are now their own chromosome and its own chromosome. So we end up with one copy going to one side and one copy going to the other side. Telophase is where the cell returns to its interphase condition, so essentially finishing up with mitosis. The nucleus is going to start reforming. Chromosomes are going to go back to their uncondensed form, so they're going to uncoil, and therefore they're going to be called chromatin again. The spindle fibers, which aren't going to be used anymore, are going to be degraded, and Temporarily, right at this point, the cells, two cells, or what will become two cells, are still attached. So for a moment in time, the cells have two nuclei until they fully separate. So during telophase, that separation is starting to happen, and these cells are going to become more and more and more separated until we actually have two separate cells. And that part is called cytokinesis. So this has already started during telophase. The cytoplasm, the organelles, remember, it's not just the genetic information that needs to be distributed between the two cells, but also all of those organelles in there. We can't have one cell ending up with no mitochondria and the other one having all the mitochondria in it. So those organelles also have to be distributed evenly between the two cells. So the cytoplasm will pinch off, and we're going to end up with two full separate cells.